We take this little tiny bit of fin and we place it inside of a tube which contains lysis buffer that breaks the cell all apart so that the DNA comes free from the nucleus of the cell. Then we use an extraction procedure that isolates the DNA and gets rid of all of the salts contained in the solution, and the very end result is pure DNA in water. And we use that DNA in water as the template for our polymerase chain reaction, or PCR reaction, which is the reaction that we use to amplify specific portions or specific regions of the DNA. After the DNA has been isolated from the tissue, we end up with a plate of 96 samples. And what we are going to do is transfer 1 microliter of DNA from each of these individual wells into a plate where we do our PCR reaction. Each tip contains the DNA for 1 fish sample. After we've loaded the DNA into the plate, we put a bunch of chemicals into a tube for the polymerase chain reaction. We add water, a buffer which protects the DNA as it goes through the heating and cooling cycles in the thermal cycler, magnesium chloride, which is essential for the reaction, the DNA nucleotides, the same ones that are in a regular DNA strand, which is guanine, thymine, cytosine, and adenine. We also add a PCR primer, which is specific for an exact region on the DNA that we wish to amplify. After we've placed all of these chemicals in a single tube, then we load the PCR plate with the master mix. This repeater pipette measures out 4 microliters every single time you push down the button. And you very gently place that master mix, which is the chemicals that will allow the DNA to amplify it in the thermal cycler, into each well top. So there's the next one. And we just keep going. Technically, we should be up like that. And I just put a little droplet on, and you tap gently, and you end up with DNA plus master mix. After I've put in all of these chemicals into the master mix, the last step is to add the DNA polymerase. This polymerase is isolated from a bacteria that grows in hot springs, and it's activated by temperature. We're basically tricking this little tube full of liquid into replicating the DNA when you heat up the master mix and the DNA and cool it. I've added the polymerase to the master mix, and now it's ready to go in the thermal cycler. So now we've loaded the PCR master mix onto the plate. We place a top to cover the wells, and then we place the plate on the thermal cycler. The thermal cycler heats up the DNA and activates the polymerase, or the enzyme that we added to the master mix. The primers that we put in the master mix, the ones that amplify that specific region of DNA, those have little bright fluorescent tags, and we use those tags to identify the size of the microsatellite as it goes through a microsatellite fragment analyzer. The PCR process is designed to amplify the DNA quickly and rapidly. You have 2 pieces of DNA that get copied, then there's 4, you have 2 more, those get copied to 8, and so forth and so on. And the DNA amplifies exponentially. Now our PCR is complete. It's time for us to transfer a very small volume from each tube of this plate into a new plate so we can put that plate on the DNA fragment analyzer. Now this robot is going to electronically transfer a little bit of liquid. This way we don't have to use the pipette by hand. These are all little teeny tiny tubes, and it goes down into each well, and it takes around 1/2 of a microliter. Now it'll go over to this one and place that liquid in the wells. This machine is our fragment analyzer, and the way it works is inside, deep inside the system, we have these capillary tubes. I'll show it on the back side. These little needles go into the wells of this plate that we've transferred the PCR product into. DNA is negatively charged, so it will go through these tubes if you have a positive charge on the opposite side of these capillaries. So the short microsatellite fragments move quickly and the long ones move slowly. The DNA travels through here, and then there's a laser that reads the color and also tells you the length of the size of the microsatellite as it passes past the laser. After samples are processed on the genetic analyzer, I use the computer to look at each individual fish's microsatellite signature, and then I use this information to determine that fish's river of origin. On the computer screen here, I have 3 different regions of the genome that contain microsatellites. Each fish has 1 microsatellite allele from a mom and 1 from a dad. This fish has different copies of each allele for each of these microsatellite markers. This is marker 1, this is marker 2, and this is marker 3.
So now we know. This fish is from the Klamath River.